Thank you, Erika, and thanks for a nice introduction. Yeah, so I have a background in food science, and I've been doing some microscopy since I did my PhD. Maybe I started more than 10 years ago, and then we're going deeper and deeper into microscopy to study food structure, and as you said, to connect with the different landscapes and the overall aim of understanding texture in food. So today I'm going to talk about a specific example with the gluten proteins microstructure, but we do much more. I will also show other examples. But yeah, I guess to begin with, I'm developing a research line here at the University of Copenhagen around food microstructure, and our idea is to connect what we understand already from food molecules, how they interact, um, to larger scales, length scales. So let's say the food structure that sits in our plate. This is relevant to us because a structure is directly related to texture. Texture is, we can say, it's the perception that generates in our brain when we destroy structure in our mouth. And so structure is a physical parameter that we can measure, but texture is not. They are related. So we want to study how these molecules build up structures to then understand texture. And this is also relevant for, let's say, destructuring processes. So we can call it mastication, digestion. But the way these molecules organize into these larger structures impacts the way they're delivered into the body. So we go from molecules to microstructures to macrostructures and then all the way down. Now there is increased interest in this part due to new trends to utilize plant proteins, for example, or more sustainable foods using more sustainable ingredients. And well, many of them, they don't create the textures that we are used to from the more traditional ingredients. So that's where we can learn a lot of the microscopic scale. The way we do it in our lab is basically we try to connect what we see in the rheology. Rheology is the study of the formation and flow of materials. So this has been well known to study food structures and food texture in a way because this is an oscillatory deformation that we can understand how the materials respond. A lot of hypotheses are drawn on how the material behaves at the microscopic level based on the rheology. And then now we're trying to actually visualize the microstructure. So we have different techniques, and closer to what Johannes presented, we focus on optical techniques. I have a review paper that's been in a long process of being published, but the preprint is available. So if anyone is interested, it's there. But basically, it's how we use more advanced techniques because in our field, people is very familiar with confocal scanning microscopy. But yeah, how can we go to super resolution? So I've used STET before to look at protein aggregates at 50 to 75 nanometers in egg white gels, for example. How can we use a spinning disk to look at the structuring process that occur in fast dynamics? Or can we go, 3D and in time with light sheet microscope or structural illumination microscopy were also used. So how can we take advantage of the different opportunities that these techniques give us to look at the food structure? And we also now focus on label-free techniques. So how we can use this coherent Raman spectroscopy microscope. We have one in our facility that can give us a lot of information about the phases of the materials without any staining. And that's one part of what we do, but most of the time I would say we spend it trying to quantify the images. And this is the challenging part right now. We're spending a lot of time because one thing is the acquisition, and if we want to correlate structure, we want to correlate, for instance, in the rheometer, there is this idea of the networks at different scales that hold the material together. So if we want to see those networks and actually quantify them, then we spend a lot of time trying to segment these images, basically, so that we can quantify the networks. So I've divided this into three main structures that we usually see in our food materials. So we can say droplets or blobs. It could also be globular protein aggregates. these networks that we see that hold the material together, and some sort of fibrillar material. Sometimes, as in the case I will show now, is how these networks can go from isotropic network to an isotropic network, and what does that mean for the texture of the material, in the case the dough. So in the review, I've put a lot of images together from, on the left is super resolution images from food microstructure. I'm pretty sure Johannes has been involved in at least more than one of these, from what I remember from reviewing the literature. And the ones on the right are only on a label-free techniques. So basically, for instance, here you can see green is the protein phase and red is the fat phase. So this has been quite convenient for us, and this is what I'm going to show today. In the case of gluten, before going to the label-free, I'm going to show what I've done before in the past. So how we're trying to look at these gluten networks. Uh, yeah, first I developed some specific antibodies for specific subunits of the gluten, and I conjugated them to quantum dots and different emissions. This equipment right here is how we may test the dough. So this is called a farinograph, and it measures the resistance of the dough to deformation. So we took samples, cryosectioning, and then we could look at the different proteins at the different stages of the mixing. This curve here is what the farinograph generates. So basically, you put water and flour, and then you mix over time, and you measure the resistance to deformation in the paddles of the machine. So we took the samples at different times, and we could look at the distribution of specific gluten proteins. And yeah, there is more there, but I just want to focus on the technique here. We also put the dough on the rheometer. Now we did this oscillatory deformation, and we got similar results. We found something new about these low molecular agglutinins and the way these networks distribute. Back then, we used some Angitool software to look at this network quantification. But this is the way we used to do it before, and that's how we connected the understanding of the molecules. For instance, in the gluten, it's well known that it is disulfide bonds that bring the network together. But it was not known how much on how molecular weight glutenin, low molecular glutenin, and by looking at this spatial distribution at the microscopic scale, we could learn new things. So now what we are doing, moving away from this, is label-free microscopy. So based on Raman, so this is a stimulated Raman spectroscopy microscopy. So basically, these two images are the same area. One is labeled with rhodanine B, and we use two photon fluorescence to look at the gluten network. But we also detect the amide 1 band from the Raman signal, and then we see how these two match. What this means is we can move on and not use the rhodamine anymore. That's in our case as a food scientist, that's important because our dough becomes pink. We never know how much. It's not stoichiometrically the amount of dyes that we add to do the staining. So if we can get rid of this labeling, we just add the dye into the water. That's the most common technique. So if we remove the dye, for instance, here we just look at the dough, no dye, and we look at the amide 1. Then we localize the proteins. We can look at the CH3 bond, and we find the starch. But we can also see the starch in the second harmonic generation signal, because the starch has these two, is divided in these two type of molecules, amylose and amylopectin. And amylopectin has this crystalline-like structure with these repetitive long strands, and that generates a very nice second harmonic signal. So we can also prove our starch there. If we look at the CH2 stretch, we can look at lipids. In case of the CH2 and the CH3, these are not the only molecules in the dough that have this stretch. These are the ones that are the most prominent. But in our case, we just wanted to see here the proteins and the starch. So here's our overlay, how it look when we look at the starch granules and the gluten network around it. And again, the fact that we can do this label-free now, it's important for us as a food scientist, because there was a lot of unanswered questions about how we are doing these stainings, also, as I showed before, then we move away from all these lengthy protocols of staining with antibodies for the gluten proteins. We look more, this is almost the same I've shown, but we also try to look at the OH stretch. So this will be in the water, so it will be everywhere. But we also added this carboxymethyl cellulose to our dough, and I'm going to explain now why. But we can also have this OH in the carboxymethyl cellulose. So in this case, we're not able to distinguish where is the carboxymethyl cellulose by following the OH because it's everywhere because of the water. So I think the only point here is that it will be relevant what other elements are in the matrix at the moment. So it's not one band or one stretch per component. It depends on what else is in the matrix. So the case for the CMC that we added, this is because we have seen in the literature that it can form these strands in the gluten network. So it goes from this more isotropic network to more anisotropic. And as I was mentioning, our interest is to understand the mechanical properties of these materials and how the microstructure is dictating those. So it's understood how this happens at the molecular level, how gluten will create these strands, but then our question is, can we quantify this at the microscopic level? And more importantly, do they affect the structural properties of the dough? So we prepared the dough again in a specialized equipment for measuring flour properties. We added the CMC, and we went for the label-free imaging. So we can do 2-photon autofluorescence and second harmonic signals for the starch, and then we're going to look at the changes in the protein distribution. How to measure the fibrils that we see in the network. First, we tried some edge detection. That was not so successful, but then our colleagues at the University of Southern Denmark, they've been working with pneumatic crystals, and they look at our images, and they thought we could use some of the analysis they do to quantify these pneumatic crystals into quantifying these fibers. So we use where they're helping us to calculate this pneumatic order parameter, and we can get the orientations in an image based on in this case on the fluorescence, and how, so it goes pixel after pixel, and it follows the pixel around that has the largest fluorescence. This is but you require a window size for this, so even requiring understanding which window size to do these measurements at it's important. In this case, the thickness of these fibrils is what determines the size of the window. And yeah, we added different levels of carboxymethyl cellulose, and we could see how until 5% we have an increase of this pneumatic order parameter, so it's becoming more anisotropic, and then there is a point in which it starts to go down. So now the next question for us is if we can somehow relate this to the mechanical properties of the dough. These are the graphs from the equipment I showed before. It's just the time and the torque. So every time we add more CMC, we see that the torque goes up, but we don't see any other significant differences. So then we took the dough to the rheometer, which is what we use the most. And here we can see we did a crib recovery test. So basically we apply crib, and then we measure the recovery of the dough, and we could see that we could gain more recovery once these fibrils were formatted, and then before and after we lose that. This is still we're still testing this because it's just preliminary, but I think the point here is when we want to connect microstructure with mechanical properties or molecular properties of foods, is we need to be careful of which mechanical properties we're measuring, because it's not all of them that will be affected. In this case, we know the CMC might be thickening the material, so we have more resistance, but we might not be able to see the effect of those fibrous formation in this farinograph or mixograph data, but in a specific type, on the specific type of deformation. So from here, so far, that I mean the message we wanted to convey is how we can detect the different components in the dough. Again, this is important for us because now we can take the dough basically directly from the mixing equipment to the microscope. If we use, for instance, only the cars mode, which works with the backward detector, we can use like an EBD slide. We can put the dough, and we don't do anything to the dough. It's just taking it directly from sampling from the mixing. To use the SRS mode, we need to use the forward detector, and that means we need to slide. So we do some cryostat, but that's basically it. Yeah, and then for our research, how we can quantify these fibrils and what does it mean for us to understand food materials beyond the rheological measurements that we're used to, and how we should keep doing more food microstructure to understand texture. That's I think this is just one example. I'm going to briefly show others later. This is the image from the autofluorescence and the second harmonic of the starch. I think this image won the image of the year here in the Danish bioimaging symposium this year. So here I just have a summary of different techniques that we have used, microscopy techniques, but the analysis method as well. And as I said, we focus a lot on the analysis method because we try to quantify structure so that we can correlate with the texture. So you've seen the gluten, we have different type of analysis. We have done some pea proteins, and we see how the networks change over time if we make gels in situ in the microscope. We're currently looking at soy protein gels. We have done this state microscopy of egg white gels, and also foams, and we try to quantify the different sizes of the droplets. This is just light microscopy. We look at the aggregation in milk proteins. So we have a heating stage in the microscope, and we add the enzyme, and we see how the proteins come together and form the structure, basically. And we also look at some hybrid foods, so they have animal proteins and plant proteins, and yeah, how they form networks and how the fat distributes there. So this is some of the activities we do with optical microscopy in my lab. And so I want to acknowledge Jens. He's the one who's been making the dough for the research I show. So he's the one who went from working with the pink dough with rhodamine to now the label-free dough. And the people at STU, Preston and Francesca, who's helping us with the analysis. Our imaging facilities here at the Frederiksberg campus at the University of Copenhagen. This is our CRS microscope that we use. Funding from Novo Nordisk Foundation, and my small team. I have 2 PhD students and a research assistant who were starting to build up this food microstructure group. So this is us, and this is us segmented. And as I said, there is this preprint of this review that you can see for more information of optical microscopy for food structure. So that's all I had for today. If there is any questions, I'm happy to take them.